So I did once stand up in front of the United Nations and give a talk in Geneva, and I was only slightly less worried about that than I am about the lecture tonight. So one hopes this doesn't go too badly. What I hope to do fairly quickly is to go through some of the ways that we can use chemical tools to understand how biology works. And I make no apologies at this stage. I'm going to actually put some chemical structures and some quite complex schemes on the board. I don't want you to remember those. I'm just going to give you a flavour. But I do understand if you start to feel slightly nauseous seeing all this chemistry, do feel free to step outside and get some fresh air. So chemistry is something that I've really been studying all my life. And I started really young. This isn't me, but it's someone who could have been me. And what it started with getting a chemistry set as a present. Now, you'd never get that these days because health and safety would be against it. But I remember playing around my chemistry set, trying to make something that went bang, or something that smelled bad, or making what this looks like, which is a very bad cocktail by the looks of things. But this is where I started. But my study of chemistry really took off when I went to school, and I had an inspirational chemistry teacher called Ron Kelsey, who used to talk about chemistry. He called it stinks, so that's where the stinks come from. And actually, back in those days, the only thing that stank more than the chemistry was his jokes, which were quite appalling, to be honest. But anyway, having sort of been brought into chemistry and been thinking that chemistry was a good idea, I went to the University of Bristol to study chemistry, and I started doing some work down there. So why would we want to use chemistry to study cells? Well, remember that basically a cell is a chemical factory. So all of the components that are in cells are chemicals. And just like a chemical factory where there are lots of processes going on, a cell is a factory for making molecules where there are complex and interconnecting processes running in parallel. And these processes are independent on each other, just as they are in a big chemical plant or a big chemical factory. But of course, because cells depend on chemistry, then we obviously need to use chemical tools to solve the chemistry problems to help us understand how cells work. So it's not surprising, really, that chemistry has a big role in biology. So having started with fairly humble beginnings understanding chemistry, really where we're going now is to follow this through to try and use many different tools, including chemical tools, but also biological tools and physical tools to study biology. So we've moved away from the idea that we just use chemistry to understand biological systems, so taking an individual discipline and a modular approach to our science. And what we're trying to do is to link these together now to make an interdisciplinary approach where we can bring together all of the science that we use, add the connectors, and get an integrated synergy where all of the sciences work together. And what I'm going to try and do over the next few minutes is give you an idea of how we can use the different tools that we have in our armory to understand how biology works. So my real sort of first study of chemistry in a biological environment was doing my undergraduate project at the University of Bristol, where I did some work on a set of compounds called the gibberellins, which are plant growth regulators. And these were discovered in Japan. When a fungus infects rice, you get this unusual behavior where the rice stem grows too tall and then breaks off because it's too heavy. It's called pecan, it's called foolish rice disease. And it was discovered that what the fungus does is it produces gibberellins, these plant growth hormones. And these are molecules, they're chemical molecules that cause the plant to grow more than it should. So I thought this looked like a really good area to do my project in and to learn something about how chemistry related to biology. I very quickly regretted that when I saw what the gibberellins were. And this just gives you an idea, those of you who are feeling nauseous, just take deep breaths. This gives you an idea of the sorts of complex chemistry that's going on. So the biochemistry of the gibberellins, these are just some of the gibberellins that are generated in the fungus that act as plant growth hormones. And this is a scheme that starts with a quite complex molecule in the first place to make all of these other molecules that are on the screen here. So this really, as I would say to the undergraduates, I don't expect you to remember all of this. I don't even expect you to understand it all necessarily. But the main thing is biochemistry uses complex pathways, even to make small molecules to affect the way that things grow. And each of the steps in this pathway, each of the chemical transformations that's taking place to turn one molecule into another, use proteins, use enzymes from biology. So they're biological systems doing chemistry. And a lot of the work I did in my early years was trying to understand how these proteins, how these enzymes work, how they do the chemistry. And that allows us to both understand how they work, but also to use or engineer them to do chemistry that we want them to do, or to do new chemistry, or block their action if we want to make drugs to stop a particular enzyme working. Actually, what I worked on in Bristol was this little molecule here. So I only got to work on a very, very small part of this whole scheme, even though I did a 2-term project. So it gives you an idea of just how complicated these things can be. Well, having had my appetite whetted for doing some work on biological molecules that have an activity and understanding how things are made, I went to do my DPhil at the University of Oxford. I went to work in the group of Professor Jack Baldwin there, and what he was looking at was the synthesis of this molecule up here. And that some of you who are familiar with chemical structures may recognize this as penicillin. So he was interested in the biosynthesis of penicillins, how are penicillins made. And the important bits of penicillins are these rings in the middle here, this 4-membered and 5-membered ring, these 2 bits fused together. That's what gives penicillin their activity. And what we wanted to do is to understand how that happens. Now, I'm sure all of you are familiar with the study of penicillin. Actually, we can probably take the study of penicillin right back to the Egyptians, who used to use mouldy bread as a poultice on wounds. And probably the mould on that bread, some of it was penicillin. If it wasn't, I shouldn't think you got better very quickly. But actually, John Tyndall first noted that penicillium, the fungus, produces something that interacted with other organisms. But we're all familiar with the fact that Alexander Fleming discovered the action of penicillin, started to understand that there was a molecule being produced by the fungus that killed the organisms around it. And this is the famous plate that Alexander Fleming had, where the mould grew while he was away on holiday, failed to wash up. It's a lesson I'm taking. Don't wash up too often. You might find something. And this grows, and what it does is you can see it killing, here's the normal bacteria, and it's killing the bacteria that are around it. You can see these little colonies of bacteria are much smaller. Now, he noted this, and from that comes, there's a very famous quote, which is one of my favourite quotes, which comes from Louis Pasteur. And it's, in the field of observation, chance only favours the prepared mind. And so he actually saw this and recognised there was something going on. But he didn't actually, so he sort of discovered penicillin, but it was a group in Oxford in the 1940s who started to produce penicillin so that we could actually use penicillin as a drug. So penicillins have a long and rich history, but the question is, how were they really made? Well, one of the reasons that I thought penicillins might be a good area is the biosynthesis of penicillins, how they're made in biology, is a lot simpler than gibberellins. It's basically 2 steps. 3 small building blocks, these 3 amino acids here at the top, are joined together to form a linear chain. So you can see a long chain in the middle here. And then in a final step, the end of that long chain is turned into those 2 rings that form the penicillin nucleus. So it's a relatively simple biosynthetic pathway. The bit that we were interested in is this bit at the bottom here. It's catalyzed by a protein. The protein goes by the very sexy name of isopenicillin N synthetase. You can say that after a while if you practice long enough. And what's interesting, though, about these sorts of things, these sorts of enzymes, is that this enzyme does this reaction here. It does it using oxygen. It uses a bit of iron and some vitamin C. It does it in water, and it does it at pH 7, at neutral pH. Now, the chemistry is to do this then sounds like it's going to be pretty simple. But actually, when we start to try and understand the chemistry, we find out that it's much more complicated. So what this enzyme is doing is it's actually closing these 2 rings. It's making 2 bonds. It's making these 2 red bonds here in order to make the 5-membered ring. So it just takes the linear chain and takes it through to the 2 rings. So that looks like it's going to be something that's fairly simple to do. But actually, almost all of the penicillins that we still prescribe are made by fermentation. We haven't got still a nice way of doing this chemistry. So nature was well ahead and still is well ahead of the chemists in terms of making this simple ring system at neutral pH with a bit of oxygen, which the chemists can't do. So we were interested to know, how does nature do that? So we started to study how these ring systems were closed. So that's that linear chain again, and what it does is it closes to give the ring system which we recognize as penicillin. So how can we study how that actually works. What sorts of things can we do? Well, what we started doing in Oxford was we started modifying the substrate. We started changing this molecule, changing the bits in the molecule at the end here. So you can see this molecule here is very, very similar, but it has a slight change at the end here. And then actually getting the enzyme to do the chemistry on these molecules and to see how those molecules behaved when the enzyme made the two rings, to see if that could tell us something about the mechanism. Well, if we take the natural substrate, the linear chain, and treat it with the enzyme, then we get one product. We get this penicillin. When we took this molecule and treated it with the enzyme, it wasn't quite so simple. This is a complicated picture. What it actually is these are nuclear magnetic resonance spectra. And each of the peaks that you can see in here corresponds with one of the hydrogens that is part of the chemical structure of the products of treating this with the enzyme. And so what we can actually do is we can use this nuclear magnetic resonance spectrum to assign the structures, to understand the structures of the molecules that we're getting. And this is what we find we get with just this simple change of the substrate. What we find is that we get the things we might expect. You'll notice this is very similar to the penicillin nucleus, a 4-membered and a 5-membered ring. So we get what we expect. But we also got what we didn't expect. We got completely new penicillins that hadn't been made before, with different ring sizes and different groups, different chemical groups around the ring. And this told us not only something about how the enzyme worked, it told us about how the different mechanisms for making penicillin worked, but also at the same time we were making new penicillins that might have new activities or new properties that would be useful to us. So this is a simple chemical tool, a simple change in the structure of the chemicals to understand both how nature does complex chemistry that we can't do in the laboratory, but also, as it turns out, to make new molecules that might have new properties that we might be able to exploit in pharmaceuticals. So that was a good lesson for me about learning how biology can do some complex chemistry that we find extremely difficult in the lab, but how chemistry can help us to understand how biology works as well. Well, having got a taste for doing biological molecules, you have to move up a bit. So I then moved from the University of Oxford up to the University of Cambridge, or at least that's what the people at Cambridge told me I was doing. And we also moved up in complexity. What I went to Cambridge to study was the biosynthesis of a very complicated molecule, this one here. This is vitamin B12, an essential vitamin that we get from our diets that we can't make ourselves. Made by microorganisms and some plants, this vitamin is a complicated, probably the most complicated of all of what we describe as the small molecule metabolites that come from biosynthetic pathways. This is made, you might imagine, by quite a complicated pathway. We wanted to understand how it worked. So here's the pathway. I hope you can all see that at the back. Yeah, you all make that out? Yeah, Simon, you're copying it down? Yeah, good, okay. In fact, I couldn't even fit it on the screen. It's gone off the bottom here. There's more of it down here, but I just couldn't fit it all on. But what's amazing is that what biology does is it starts right at the top here with a molecule that contains 5 carbon atoms, just five carbon atoms in a chain. And it links them all together through a whole series of different steps to make vitamin B12, which has over 60 carbon atoms in it. So it joins together a number of units to make vitamin B12, and so it takes 5 carbons and turns it into 60. So we were studying this pathway and how it worked. Now, this was in the 1990s, and what was surprising is we thought we knew everything in the 1990s, but we really didn't quite know how this worked. The mechanism, or the biosynthetic pathway, was elucidated in the early 1990s at the time when I was working on this in Cambridge. And what was really nice about that was that for many, many years, the pathway, how it worked, had been talked about by some of the greatest chemists that were around at the time, Albert Eschenmoser and Professor Battersby, who I was working with at the time. And they'd drawn a scheme of how it would all work. They had it all worked out in their mind exactly how vitamin B12 would be biosynthesized. And they had it wrong. When we actually found out when this molecule here was isolated as part of the biosynthetic pathway, it showed that the mechanism that they decided it would work by was wrong, and that nature had done it a completely different way. And of course, as soon as they saw that, they said, it's obvious that's how nature should have done it. We can see how it works now, yes. But what was amazing is it it did, even in the 1990s, the greatest chemical minds couldn't sort out how nature had done such a complex bit of biochemistry. So anyway, having elucidated the pathway, we were interested in then looking at a bit more what went on. We actually went right back to the beginning here, and we started looking at how this molecule here is made in biology. So we'll just have a look at that in a bit more detail. What actually goes on at the start there is 2 of those 5 carbon chains are joined together to make this molecule, which has 10 carbons in it. The next enzyme, the one that we were interested in, takes 4 of those and joins them together into a linear chain. So you can see there's 1, 2, 3, 4. So this is the enzyme we were interested in. It has another one of those really sexy names that you just roll off the tongue, hydroxymethyl bilane synthetase. Yeah, biologists aren't very good at thinking up names, are they, when you get down to it? But this is the enzyme we were interested in. And what it does is it takes 1 of 4 of these and it joins them together in a linear chain. We were interested in this because this is the start of a number of very important pathways in biology. Because the next thing that happens is these 4 groups are joined together by joining one end here to the other to make a circle, to make a macrocycle. This is a cyclic compound. You can see it's a complete circle. And that's actually the precursor for a lot of very important bits of biology, these molecules over here. If you look carefully, you can see where this circle occurs. It's the red here, the green here, and the purple here. So you can see that this intermediate is present in all of these different molecules. So what are these molecules? Well, the one at the top here is heme, from hemoglobin, that carries oxygen around your blood. The green one here is chlorophyll, the thing that is involved in harvesting light to grow plants, which is responsible for almost all of the energy generation on our planet. And the one at the bottom down here you've seen before, that's our vitamin B12, our incredibly complicated vitamin. So this intermediate, this critical first few steps, generates an intermediate that's really important in synthesizing some very, very important biological molecules. So we were interested in understanding how this takes place. So how can you actually start to work on understanding how a protein puts together a chain like this? Well, we decided to use what was at the time a relatively new technique to be used in interface with biology. And it was a chemical technique that had been used for many, many years to study chemistry, but we thought we could apply it to biology. And it's a technique called mass spectrometry. Now, mass spectrometry is a technique that had been used by chemists for many years to measure the weights of molecules. It's able to measure the weight of an individual molecule or a collection of individual molecules so you know how much those molecules weigh. And chemists have been using it because if you can do that accurately, you can actually identify what the molecule is just from its molecular weight. And also, by breaking up the molecule in the mass spectrometer and measuring the weights of the bits that it breaks up into, we can say something about the detailed structures of those molecules as well. So mass spectrometry was a really useful tool in chemistry. Another thing that it did, it was a very sensitive technique, and I'll come back to that in a minute. So we were interested in seeing whether we could apply this to a biological problem. Now, of course, it's very nice to be talking about mass spectrometry here at the University of Aston, because this is Francis Aston's mass spectrograph, which he built in 1919 in the labs of Thomson in Cambridge. And this is probably the first ever proper mass spectrometer, and it was built by a guy called Aston. Now, of course, where we are now is in Aston, and this is where we are in 2012. This is one of the mass spectrometers that we have in our labs here, being beautifully modelled by one of my collaborators, Dr. Corinne Spickett here. And so we've moved quite a long way from Aston's mass spectrograph, but it's nice to be here in Aston doing mass spectrometry. So what does mass spectrometry do? Well, it allows us to identify things by mass, and it allows us to do some quite complicated things. So using mass, it's fairly easy. So these are some of the group of my collaborators in Glasgow. It was fairly easy to tell Logan here, who's a big strapping chap, from Stefan here by mass. Stefan is a vegan, so he's always been a little bit weedy. Can I say that? Yeah, okay. So it's pretty easy to tell those two apart by mass. So just by weighing them, we'd have known which one was Logan and which one was Stefan. It can get a little bit more complicated than that, because Inge and Jay weighed about the same, but actually they're quite different, as you can see. And it's only by looking at them in detail and seeing what they look like that we can actually tell that they're different. So we wouldn't be able to do it by mass alone, but if we could get some more detail, then we'd be able to tell whether it is Inga or J. Now this is made even more complex in proteins because protein modifications take place. That is, the protein can be modified, and proteins can be modified by having small additional units grafted onto them. I'm going to come back to this a little bit later on. But what this means is that we get a very small change in mass that might be quite hard to see, but we can have a very big change in function. So here's Kat, and sometimes, this is Katrina Thompson, one of my group in Glasgow, and sometimes she used to graft little bits on when she was having a bad hair day. Clearly, weighing her, we might not be able to pick up that she's changed, but clearly the change that's taken place has been quite significant. So how can we use this to study biology? Well, we were interested in studying this pathway, and we were interested in studying the enzyme that put together this chain. So what did we do? Well, we took a mass spectrum of it. We gave the enzyme, the protein, some of this, and we took a mass spectrum. What we got was quite a complicated picture. And if we look at this in detail, what we can do is we can discern something about what's going on in the chemistry on the protein from the detail of this mass spectrum. So this is just a little bit of that mass spectrum, just one of the sets of peaks in it. And what you can see is there's a number of different peaks in this mass spectrum. So the first one here, we can work out what the molecule weighed. And we know what the protein is. So this is just a representation of the hydroxymethyl bilane synthase, the protein we were interested in. And we can calculate how much that should weigh. And what we found is that what this should weigh didn't correspond with what we actually measured. So we had a problem. So we had to think about what it was. It wasn't very much. It was only a small difference. And by working out the difference in mass, what we could see is that actually we already had two of the units that go to make up that four-membered chain attached to the protein. So when we'd isolated the protein from its natural source, it already had two of the units attached to it. That was a bit of a surprise. We didn't think it would have, but the mass spectrum told us that was the case. So that explained this first peak. When we looked at the mass difference, the second peak, it became fairly obvious what that was. It was that another unit had been added to the end. So there's now 3 in a row. And when we moved to the next peak and worked out what the protein weighed, it was fairly easy. We had 4 in a row. So that's great. There's our chain of 4 units added together. So that was our product that we were looking for. So that's fine. That's what we expected. But you'll notice that's not the last peak here. And the difference in mass between these two is another one added onto the end. Well, hang on, that's 5 now. And obviously, nature can't count. And it's even worse than that. The one at the end here is actually 6. And then we realized what was happening is that actually, for some reason, the first 2 molecules are attached to the protein, and they stay there. A chain is built adding 4 more molecules on to make a chain of 6, and then the end 4 of those are cleaved off, are broken away to make our linear chain, and the 2 that we started with stay there. So it was a complicated bit of chemistry. We now understand why the protein does that. I'm not going to explain exactly why, but we could now understand it. But we only started to understand it when we could unlock what was going on at a molecular level on the protein and how it was making this linear chain. And it was a bit of a surprise to find out that it put 6 together in order to make 4. So 2 plus 4 is 6, minus 4 is 2 again. Nature can do maths. Good. Okay, so that's the way that we could apply mass spectrometry to understand what was going on in biology. Now, I mentioned that mass spectrometry is a very sensitive technique, and that's very good because it allows us to look at relatively small biological samples. So current instruments, current mass spectrometers, the ones that we have upstairs in this building, can measure 10 femtomoles of compound. Okay, who knows what 10 femtomoles is? Yeah, me too. That was my thing. What? A what? Okay, so I want to try and give you an idea of what 10 femtomoles means. So there's a little table down here showing you what we go through. We've got moles here, and what a mole is it's the number of molecules that corresponds to 12 grams of carbon. It actually turns out to be quite a lot of molecules. It's 6 times 10 to the 23 approximately, but it's 1 mole of carbon corresponds to 12 grams of carbon. And what this allows us to do, because that's a fixed number of molecules, it allows us to compare different chemicals not by their weights, but by the number of molecules that we have present. So a mole of hydrogen weighs 1 gram, a mole of carbon weighs 12 grams. Now here's a mole over here, and here's femtomoles. It's 10 to the minus 15 moles. So 1 mole of carbon weighs 12 grams. If we divide that by a million, then we get down to approximately where we want to be. Well, numbers like that do my head in. So what does it really mean? Well, I'm going to try and illustrate it for you. Everybody know what this is? Yeah, bag of sugar. How heavy is that? Kilogram. Okay. So a kilogram is a kilogram of sucrose. So that's approximately 3 moles of sugar we've got in here. Sucrose has a molecular weight of 342. So it's approximately 3 moles of sugar in this bag of sugar. So let's then take a grain of sugar from this bag. How many moles are in that? Well, first of all, what we need to know is how many grains of sugar are there in a bag? Well, in here I've got a very dangerous grain of sugar. Can everybody see that? Yeah, okay. So how many grains of sugar are there in a bag of sugar? Take a guess. Millions. You wouldn't believe this. Someone's actually bothered to... No, they didn't count. They calculated, all right. They didn't count them all. And it turns out to be about 20 million. Okay? So it's about 20 million. The numbers vary, about 17 million probably, but 20... I'm not very good at maths, and it's a nice round number. I can do 20 million. So in a bag of sugar, there's 20 million grains of sugar. So if this is 3 moles, this corresponds to 150 nanomoles. So we're down here, 10 to the minus 9 moles. It's about 50 micrograms, micro being down here, 10 to the minus 6. So it's about 50 micrograms of sugar in 1 grain, about 150 nanomoles. And that's 1/20 of a bag of sugar. So what's a femtomole? Well, a femtomole is effectively a 1/20 of that. So if we take our grain of sugar and break that up into 20 million bits, then we'll be down at a femtomole of sugar. And that's how sensitive mass spectrometers are. They've become extremely sensitive. And when people bring me a sample and say, is there enough? I say, if you can see it, it's more than enough. We can now work with very, very small amounts of material. This sounds like very small amounts, 7.5 femtomoles of sugar and 2.5 picograms down here. But if you remember that a mole is 10 to 23 molecules, this is still 100 billion molecules of sugar. So maybe we're not being all that sensitive at the end of the day, but it certainly is a very small amount indeed. So how does that become useful? How can we actually utilize the sensitivity? Well, how useful is that sensitivity? Let's think about one cell. If we wanted to study one cell by mass spectrometry, how small is a cell? How much material do we get from one cell? Well, a typical mammalian cell is about 10 to 20 microns in diameter. That means you can get 1,000 of them across the width of your finger. Okay? So if you've got a pen on, you try drawing 1,000 lines across your finger. Okay? That'll give you an idea of how small they are. Okay, so what does that mean in terms of the amounts that we've got? Well, it has a volume of about 1 picoliter. So that means you only need 100,000 million of them to make a cup of tea. Okay? 100,000 million cells in a cup of tea. It may not be a very nice cup of tea if you make it from cells, so I wouldn't recommend it. But you can see cells are really very, very small in volume. And what that means is that they only contain very, very small amounts of material. So we've got about 50 picograms or 2 femtomoles of total protein in a cell. So if we're working with very small amounts of biological material, we do need extreme sensitivity. And in 1 gram of a dried wheat meat, I had a South African working in the lab. Everybody's had biltong, I wouldn't recommend it. But dried meat, in a gram of dried meat, there's about 1,000 million cells. So that gives you an idea of the sorts of sensitivity we'd need to have in order to be looking at small amounts of biological material. And within each individual cell, there may be only 10 molecules of a protein that we're interested in. So we may need 10 million cells in order to do one experiment on the mass spectrometer, even though we have the sensitivity that we have with the mass spectrometers in the laboratory we have now. So sensitivity is important if you want to go down to look at small samples in biology, but we're still a long way off being able to do a single cell by mass spectrometry. Anyway, so that's the technique that we're using. What sorts of things that can we use it for? Well, one of the studies we've been doing is to use it to look for things called biomarkers. Now, biomarkers, the definition is it's a measurable indicator of a biological state. So something that we can measure that tells us a biological state. Now, you're all actually pretty familiar with biomarkers because a biomarker may be an echocardiogram of your heart to see what your heartbeat is like, or a measure of your blood pressure. Those are biomarkers. They tell you something about your physiological state. They may be genetic. We all know that the human genome has been sequenced, so we may be able to use genetic markers to give an indication of people's propensity for disease. They may be more observational biomarkers. So, did you have too much to drink last night? Or what we're more interested in is the molecular biomarkers. Can we find molecules that give us an indication of a disease going on? Now, a classical molecular biomarker that you're all familiar with is glucose. The levels of glucose in your blood can be used to give an idea of whether you're suffering from diabetes or not. So that's a molecular biomarker. But what we want to do is we want to try and develop on molecular biomarkers that can be used to characterize a number of different biological processes. First of all, to define what's normal. I know that sounds like it's easy, but actually at the biological level we're not very good at knowing what's normal. And then we need to try and link that to understanding what's going on in pathology. Can we find molecules that tell us about a disease, whether someone has a disease or not? And then, if we can we use those same molecules to see how the drugs that we're giving them are improving that state? So that's what our hunt for molecular biomarkers is doing, and we're using mass spectrometry to do that. We were in Glasgow. Glasgow is a very good place to study cardiovascular disease. You can see where Glasgow ranks on the table of cardiovascular disease events, coronary events per thousand, right at the top of the European scale. So when I was up in Glasgow, it was a good place to be studying cardiovascular disease, so we started looking for cardiovascular biomarkers. So why is Glasgow a good place to be doing cardiovascular research? Well, this is breakfast, okay? A nice, good fried breakfast, nice pint of beer at breakfast time just to warm you up for the day. This, of course, is the well-known delicacy, the deep-fried Mars bar. Yes, you can get deep-fried Mars bars in Glasgow. So maybe the diet, the culture, and the weather are something to do with very high risk factors. Metabolic syndrome, inflammatory disease. Glasgow is a good place to be studying these. Also, they had some very good banks of samples that we could use to try and find biomarkers that were related to cardiovascular disease. And the method we used to do this was using mass spectrometry. Now, this is a complicated picture, but basically each of the little spots on here represents a protein in urine. So we were taking urine from people and we were measuring the amounts of different proteins in the urine. And what you can see is that each of these spots, each of the intensities of these spots, tell us how much of that particular protein there were in their urine. And we can do that using mass spectrometry. And you can see this is just a little slice out of the middle here. We've got thousands of individual spots in here that we can use to try and find markers that are related to disease. So we can take these, we can do complex statistical analysis, and we can identify markers that are related to disease. And we use this to try and study cardiovascular disease, and it turns out that we can do it reasonably well. We found that if you take a panel of 15 of those spots, we can actually, with reasonably good statistics, I'm not going to go through the statistics, tell whether someone has a risk profile of developing cardiovascular disease in asymptomatic people. So we can take someone who's not showing symptoms of heart disease, and using these 15 markers, we can tell people their risk of developing cardiovascular disease. So, by the way, this is the people with cardiovascular disease numbers, the mean value of F. This is just a classifier that we use. NED here, anybody who knows Glasgow, in Glasgow, NED stands for non-educated delinquent. So this means no evidence of disease here, okay? These are not the NEDs from Glasgow. But what we could do with this panel of biomarkers is we could tell people their risk of developing cardiovascular disease, and we could monitor their progression after treatment by the changes in these molecules, how the therapy was improving their cardiovascular disease. So mass spectrometry has uses outside just understanding the mechanisms of biology. We can actually understand some population biology using the sensitivity and the resolution of mass spectrometry. So it's a very powerful technique. We can also use mass spectrometry to make pictures. This is how we do it. We take a bit of tissue. We take a mass spectrum from a range of, each place across tissue. Each one of these spots is somewhere where we've collected a mass spectrum. And from all those mass spectrum, we can plot the intensity of individual signals across the tissue. We get a picture then of the distribution of that particular molecule across the tissue. So this molecule here has this distribution across this slice of brain tissue. So we can actually use it now to make molecular pictures and see if we can use those molecular pictures to understand disease. Well, one of the diseases we looked at with this was stroke. This is a magnetic resonance image. Now, a little bit earlier on, I talked about nuclear magnetic resonance spectroscopy when we were looking at the products that we got from treating the enzyme in penicillin biosynthesis with a new substrate. This is actually another application of that methodology which was developed by chemists for looking at the structures of molecules. This is the same methodology being used by medics to look at the structure of people, in this case animals. So this is another thing that's actually another chemical tool that's being used in biology. But we were working in collaboration with people who were looking at stroke, and this white area here corresponds to an area affected by cutting off the blood supply to the brain. So this is a region of stroke. And what we could do is we could use our imaging using mass spectrometry to look at the molecules across that region. And these are the sort of pictures that we get. We find some molecules which are across the whole brain. So this is a slice through the brain in the same way that these are here. And these molecules are across all the brain. But actually this molecule here disappears in the stroke region, which is down at the bottom here. And this molecule here appears in the stroke region, which is at the bottom here. But we have much more molecular detail than that. We can look at thousands of molecules doing this, and we can find other molecules that actually occurred right in the middle of the stroke region in the brain. And what we were able to do using this is to identify that this tissue was necrotic. It was dead. That was already dead tissue. There was nothing you could do about it. There was no way of recovering that. We could find that this pink tissue here was going to die. So this is tissue that was already so badly damaged that it was going to die. But around the edge, we could identify tissue that would survive if the stroke victim received the blood flow back to that stroke region. We could generate three-dimensional images of that, so we can actually look at the volumes of brain that correspond to each of these regions. And by doing that, what we could do is we could start to look at drugs that would protect the brain from damage after a stroke by seeing how the volumes changed, how the regions of brain that could recover were improved by the drug treatment, using, in this mouse model, our mass spectrometry to map regions of the brain that will never recover and regions of the brain that stood a good chance of recovering. So here we have now another chemical tool, mass spectrometry, being taken right the way through to generating medical images that allow us to understand how we might treat, or how treatments might work on stroke. Now, you can actually use this method to do a whole range of things. We can look at tumours using this mass spectrometry imaging, making pictures from mass spectrometry. And we can say something about how drugs improve the chance or break down the tumour and remove the number of tumour cells. One of the studies we did, which I always found a little bit strange, was this one here. This is schizophrenia, looking at for molecules that change in the brain in schizophrenia. This is PCP-induced schizophrenia. I think most of you will know what PCP is, and it's a common thing that's been observed in humans, is that if you take PCP, some people develop schizophrenia. Now, we're not allowed to cut people's brains up to do the work that we do. Somehow people are very protective of their brains. But we could treat mice with PCP and look at their brains to see if we could find molecules, and we were finding molecules that change when they were treated with PCP. What I never was quite sure was how our mouse model, how can you tell it's schizophrenic? Does it think it's a rabbit? I never was quite sure how they did this. But anyway, we were able to use the mouse model, and we were able to identify molecules that are related potentially to the changes in the brain that takes place in PCP-induced schizophrenia. So that's a whole range of chemical technologies or chemical tools being used to study biology. We were also interested in how cells sense their environment. This is a white blood cell chasing a bacterium. This little black blob in front of it is a bacterium. And it's chasing this bacterium as the bacterium tries to swim away. So this cell is sensing where that bacteria is, and it chases it until it captures it, and then we all know that white blood cells capture and destroy bacteria. So we were interested to know, how do cells sense their environment? Could we take chemical tools to understand how this happens? Well, we know a little bit about how they sense their environment. What cells have is, around the surface of the cells is a membrane, and in those membrane are embedded proteins, and when molecules from outside the cells bind to this protein, a signal is transmitted across the membrane and into the cell, and usually into the cell nucleus to generate changes in DNA. So here's a fairly typical picture. This is a signaling pathway. It's called the Ras-Raf-Merkirk pathway. But the main thing to take away from this is, if we have a signal such as a growth factor affecting a cell, what that does is it activates the receptor on the cell surface. That receptor then recruits proteins from inside the cell, having been activated by this external signal, and that signal might, for instance, be the signal coming from the bacteria. Here we have then, what takes place is a very small change. A few little bits of decoration are added to this protein, but that decoration, its phosphorylation, the addition of phosphorus or phosphate to the protein, activates it. What that does is it gets this protein then to activate the next one down, and that one activates the next one down, and this one activates proteins in the nucleus called transcription factors that turn on gene transcription. So that's how cells effectively sense their environment. An external signal binds to a protein on the surface, a set of events, a signaling pathway takes place from the changes that take place on the binding of the molecule to the receptor inside the cell, and that causes changes in gene transcription. So that's basically how cells work. But what we found, or what we know, is that this pathway, these proteins here, are involved in a whole number of different extracellular signaling events. It might be signals for proliferation, for growing, or for cell cycle to turn on changes in the cell, for differentiation to turn into different cells, transformation, survival. So just to illustrate one of those, this is a model of decision making in the cell. How does the cell decide what to do? So we treated cells with EGF, epidermal growth factor. And what epidermal growth factor does is it causes these cells, they're called PC12 cells, there's a special sort of cell, it causes them to proliferate, to grow. If we treat them with a different signal, a different extracellular signal called nerve growth factor, what these cells do is they differentiate into neurons. So if we treat them with one extracellular signal, they proliferate. If we treat them with a different signal, they differentiate. So they undergo completely different biological processes. But they use a single pathway, this signaling pathway that I just showed you, to take those two signals into the cell and take them to do the different events. We know now why that occurs. In this pathway, we get a transient activation, which causes proliferation, and in this pathway, we get sustained activation of the pathway, which causes differentiation. But how does that work? How can one simple pathway take different signals and give different results inside the cell through one route? Well, of course, what it means is that it's not as simple as that. It looks like a nice linear pathway that's taking place in the cell. But what we know now is that that's embedded in a complex network of other events, and it's all of these other events that are responsible for that change in the pathway in the cell. So how can we study that? How can we understand what's going on? How can we understand how this myriad of signals can have all of these different effects on the cell? Well, there are a whole lot of ideas about how that might happen. How can we study what goes on? Well, what we use is a process called systems biology. We start to model the system to understand how a cell works. And we take complex pathways and dynamic behaviors of cells and try and understand how those happen. But how can we measure those sorts of things? Because the biology is pretty tough. We've got thousands of events taking place. The technology, well, do we have the technology to be able to cope with that biology? Well, we decided that we didn't, and that we needed to design something that would allow us to do this. And actually, what's going on in here is not that much different to an electronic circuit that's taking a number of inputs and giving a number of different outputs. And so what we developed was something that we call the molecular nose. Okay? So we're back to stinks again. So chemistry goes full circle. We've developed a methodology called the molecular nose. And what does this molecular nose do? Well, cellular processes depend on this thing where we get an extracellular signal that activates something in the cell membrane. We now know that we have a complex pathway inside there where many things interact with each other. That causes changes in gene transcription within the cell. Then there are lots of other events that take place as a result of that gene transcription, which result in a very, very complicated picture that we were finding it extremely hard to model. So what we decided to do was to change that. We decided to design our own intracellular system where we'd get the same effect on the cell surface, the same complex biochemistry taking place, but we'd introduce a sensor which would measure directly the output of those signaling pathways. So now we're reducing the complexity inside the cell, we're reducing the complexity of what's going on, and we can get a straightforward readout of the activation of these pathways by the different signals that we have here. And then we can generate a model that will help us to understand how all of those complex pathways interact with each other to generate the effect within the cell and explain why one signal gives one effect, another signal gives a different effect through the same pathway. And this is the sort of readout that we get. We can get different activations of different transcription factors. Remember, I talked about transcription factors earlier on. They're the proteins that are responsible for turning on gene transcription. And what this allows us to do then is to build a complex model of exactly how the cell is working. And you can see these models can get very, very complicated. But now what we have is we have a few receptors at the surface here which get activated, and we can understand how all of the different events downstream of that take place by the individual activation of those receptors. Why did we call it the molecular nose? Well, the way that your nose smells things, how it senses smell, is there are a number of receptors, those cell surface receptors, the same sorts of things. There's a few hundred of them, different ones in your nose. But from that, you can smell thousands of different smells. You can distinguish maybe 10 or even 20,000 different smells. And how that works is your mind, your brain, integrates the signals from those small numbers of receptors and is able to distinguish the smell of grass from the smell of apples. And what the system we have is doing is it's taking those signals and interpreting or measuring the signals that come from the cell surface inside the cell, measuring those signals so that we can deconvolute the pathway and understand how this complex biology leads to its end result. And of course, this is important because one of those pathways is the growth factor pathway. It's the pathway that's involved in controlling cell growth. And if you have uncontrolled cell growth, you have cancer. So this helps us to start to understand those processes. So hopefully what I've done in the last longer than I intended is tell you something about how we can use a range of different chemical tools to unlock biology. We can use synthesis of molecules to understand mechanisms. We can use mass spectrometry to measure what's going on. And we can use things like the molecular nose to deconvolute complex biological processes. So with these tools, we're better armed to fight disease. Any tool is a weapon if you hold it right. What we have now, because chemistry has added lots of tools to our armory, is we have our Swiss Army knife. We've moved away from the days of the Russian Army knife to a more complex environment where we can use many different techniques to understand biology. So really that's the message I wanted to give you. That's everything I have to say. I should thank people. There are too many people to thank individually. So what I want to do is to thank the many people throughout my scientific career who've both helped and inspired me to end up doing the science that I'm doing now. All the people that have worked in my research groups and my collaborators who have done the science. The funding agencies who are responsible for giving us the money for actually being able to do the science, and the charities who do that as well. So this is a typical Friday afternoon in my laboratory here. You'll recognise it if you look in through the window. Is we can apply these chemical tools now to understand biology. I'm going to leave you with just one other quote, because I like quotes. So you saw the one from Louis Pasteur earlier. This one is attributed to Albert Einstein. When I was working in Cambridge, an Australian postdoc arrived in the lab. He arrived with a poster tube. He unrolled a poster and he stuck it to the wall. And this was the first day he arrived. And what it said was, if we knew what we were doing, it wouldn't be called research. So I've taken you through an odyssey of chemistry applied to biology, but at the end of the day, we're still learning. We still can't answer all of the biological problems, and we're going to continue to apply chemical tools to understanding biology. So I hope you've enjoyed that. Thank you for listening.
Thank you, Andy, for taking us through, somewhat sympathetically, through the complicated chemistry. You were very gentle with us. So I'd like to open that up for anybody who'd like to ask Andy some questions about his work.
They all know there's wine across the corridor. So.
Some of the mass specs that you showed up, was it all the same technique or were they different techniques?
Actually, the mass spectrometry I showed, you might have noticed that it looked like the picture I showed of the peaks was blue with a white line on it. That was actually a copy of a slide that I had made when I was doing this work in Cambridge, which is quite a few years ago now. I'm not going to say how many. And in those days, we had the first mass spectrometer of its type. It was called an electrospray mass spectrometer, which allowed us to look at these big biological molecules, which were really not very strong, so they break apart very easily. And it was a very soft way of measuring the mass of them. So that was my first introduction to this new technique, which had just been introduced in the mid-1990s, called electrospray mass spectrometry. And that's actually the method we use for most of our mass spectrometry today, is that same technique that we brought forward since the 1990s. The mass spectrometers themselves have got more complicated. We can now measure mass much more accurately. We can measure even smaller amounts of samples, down to attomoles. Everybody remember where attomoles were on that? Yeah, somewhere below femtomoles. They're more sensitive, but basically they're the same machines that we were using in the 1990s.
I think I'll add one note. Maybe I'll take it back. I guess biomarkers is one end, and a lot of people have been putting great faith on personalised medicines. How wide a gap do you still think we're at?
Well, what you have to remember about personalised medicine is people think personalised medicine means you're going to invent a drug for me. Okay? Now, some of you may know how much it costs to make a new drug. It's running at about $1 billion now. Now, I know I'm worth $1 billion, but I'm not sure all of you are. So what personalised medicine is not about is making individual drugs for people. What personalised medicine is about is making sure that we take the appropriate drugs and treat the appropriate patients with those drugs. So it may be that certain patients may not respond to drugs. It may be that certain patients will show side effects to drugs. If we can find biomarkers that will inform us as to what's an appropriate drug to give, in what dose, in what sort of regime, then we'll move away from, if anybody who's been in hospital and been on any of these drug regimes of this thing, we'll try this for a few days, and if it doesn't work, we'll try something else, to actually being able to prescribe the right medicine at the right time. And of course, what that will do is not only improve the experience of the patient, but it will save the National Health Service millions and millions of pounds in wasted medicines.
Unless somebody's already asked a question about the mass spec, but sort of the mass specs that you look at in chemistry, obviously just lines on sort of a graph. How do you get the 3D images? Sort of like the sweeping, is that to do with?
Okay, so there's always a smart guy at the back who asks the difficult questions. You know. There's a bit of a trick in that. So what we can do is we can take a slice of tissue, and we can take a spectrum at each point in that slice, and we can generate that two-dimensional map relatively easily. What we then have to do to generate the three-dimensional maps is we have to keep slicing through the tissue, taking slices at various places. So now what we have is lots of two-dimensional images, and we can use software to reconstruct that into a three-dimensional image. So it's a bit of a cheat. We can't actually do it in 3 dimensions, but I'm allowed to lie. I'm a professor now. So yeah, good question.
What's next in your research?
That's more of the same. Well, so I guess the message that I put up at the end there, which was if we knew what we were doing, it wouldn't be called research. I started working in cancer about 20 years ago. I went to a conference in Killarney in Ireland, in a little place out in the back of Ireland, just as these signalling pathways were starting to be discussed. And I went and people were drawing that linear pathway that I showed you for the MAP kinase pathway. And I thought, this is great. We now know how it all works. And all that we've done since then is proved every year that it's actually more complicated than we thought it was. And so I say this to the students. I now probably know less than I did 20 years ago about cancer, because we know so much more about how these signaling pathways work. So if we'd solved all the problems, then I'd be thinking, what am I going to do tomorrow? But actually, what we're doing is it's not more of the same, it's expanding what we have now, trying to make better models, to make better technologies that allow us to go deeper with less sample and understand more about this complex biological environment we're working in.
One last one.
Can you say what is your driver, following on from the previous question? Is it the medical need, pharmaceutical companies to get better drugs or whatever, or is it your passion for the chemistry, biochemistry to drive it, or a bit of both?
Oh, it's the money. It's the no. So, when I was at Oxford doing my PhD, I had lots of friends, and half of them went on to do science, and the other half went into the city and did banking. They're now multimillionaires, and I'm here at Aston, not quite a multimillionaire, but I keep buying my lottery ticket. You never know. So, the driver for me is the science. I mean, at the end of the day, if you look at where I started studying plant growth hormones in gibberellins in Bristol, doing chemical synthesis, and I now have a lab where we do technology development on mass spectrometers. We do biology, we do a bit of physics, we do some chemistry. It's science. It's the richness of the science environment. It's the fact that every day I learn how little I know. It's every day we make one step forward and we unlock another Pandora's box of biology. And the challenge of eventually we will understand how all this works. And if we just keep moving forward, and if we can, there are days where you just feel like giving it up and going home because you're not making progress. But every day we make a little bit more progress. That's the driver is just a fascination with how biology works.
You're not telling the rest of that.
Oh.
Yeah, I never told Andy enough, and I know there's a nice buffet waiting on you there. So thanks again for that.
So I have to say thank you very much. I have a question actually, which is penicillin a chair-shaped molecule?
Yes, it is slightly.
Because I remember that I gave up chemistry in my second year as an undergraduate and became an engineer because we'd had about 70 lectures on organic synthesis, and we got to the chair-shaped molecule, and I thought, oh my God, is there a table-shaped molecule? And I thought, where is this going? And nobody ever told me that it might have been penicillin. It might have been more interesting.
It's shaped more like a psychiatrist's couch than a...
But I think I have to absolutely thank you for giving us the definition of an academic researcher, which is somebody who can spend £16 million and at the end of it can say, I now know less than I did. So thank you very much for the super lecture, and here is a small